Welcome, everybody, to this lecture on the topic of agonist efficiency. This is going to be an extended version of a talk that I gave about a month ago on May 23rd of this year at a conference on the biophysics of ligand-gated ion channels held in Ottawa, Ontario. So in the most broadest sense, agonist efficiency relates to understanding how agonists turn on receptors. So if you don't know what an agonist is or what a receptor is, this talk is probably not for you. But if you do know, you may want to stay tuned because I think agonist efficiency lies at the heart of understanding how agonists activate receptors. This video pamphlet is divided into three sections. This is the preface, in which I will try to put efficiency in a nutshell, trying to describe why it is important and general. And then I will follow with a few slides on nicotinic acetylcholine receptors. If you're familiar with these receptors, or you just don't care about the protein and you can't wait to get to the main topic of agonist efficiency, I encourage you to skip that section and go straight to the second section of the video pamphlet, which is on agonist efficiency per se. That section is divided into 4 chapters. The first chapter gives definitions, and I'll start with some background before I give the definitions of affinity, efficacy, and efficiency in terms of binding energies. And I'll briefly consider the difference between single channel and wholesale current measurements. The second chapter is about patterns of agonist efficiency in nicotinic receptors. And I will start this chapter with what I'm calling a surprise, an unexpected experimental result. And that is that there is such a thing as an efficiency class. Many agonists have the same efficiency. This is really what makes agonist efficiency a very interesting parameter. And the third chapter is about the origins of agonist efficiency in terms of energy landscapes. And I will again start with an unexpected experimental result, this time with regard to the agonist association rates constant. And at the end, I'll very briefly consider other receptors and some potential applications of efficiency. And in the fourth and final chapter, we leave the agonist and we turn to the rest of the gating isomerization. And I will again start with an unexpected experimental result, this time with regard to a parameter called phi or phi, which gives the energy of the transition state at different regions of the protein. And I will end this section by trying to link the binding and the gating parts of the agonist activation process into a zipper mechanism. I hope you'll stay around till the end when I will give an extended acknowledgement sections, but in case you don't, I just want to briefly mention the 5 people who I think were the main players in the agonist efficiency part of this pamphlet. Snehal Jayati, who was a graduate student, Prasad Purohit, Eva Bruhova, Tapan Nayak, and Dinesh Adurthi were all postdocs in the lab and made major contributions to the project. So why efficiency? So here's agonist efficiency in a nutshell, or more specifically, 4 different nutshells. First, what is agonist efficiency? It is the correlation between affinity and efficacy. And insofar as every agonist has an affinity and an efficacy, every agonist at every receptor also has an efficiency. So agonist efficiency is a universal attribute of agonists and receptors. The second reason to study efficiency is because it gives you the strength of the energy link between binding and gating, gating being the receptor conformational change. And insofar as that link is the essence of understanding how drugs activate receptors, I think agonist efficiency is a very important agonist parameter. Number 3, it is organized. It has structure, meaning it is patterned, and anything that is patterned has information. So it is informative, and that's what we're trying to find out is the information that it's telling us. And the fourth reason to study agonist efficiency, because it is the trigger for the allosteric transition of the receptor. And in that sense, what we learn about efficiency we think can be extended to other aspects of receptor activation. So next, I want to introduce to you the nicotinic acetylcholine receptor, which is the preparation that we have studied. It is a ligand-gated ion channel that has 5 subunits that surround a central ion permeation pathway. It has a discrete extracellular domain, transmembrane domain, and not shown here is an intracellular domain, which is disordered. In the extracellular domain, about halfway up and at subunit interfaces, there are ligand neurotransmitter binding sites, which are the sites that we're going to be concerned about today. And in the at the equator of the transmembrane domain, there's a narrow region in the pore called the gate, which regulates ion conductance. Total molecular weight of the protein is about 300 kilodaltons. Now, nicotinic acetylcholine receptors are allosteric proteins, which means they have 2 global stable conformations that each have a different output and a different input. The different outputs are off and on, and different inputs are weak or strong. And since they are ion channels, the off and on refer to whether the pore can conduct ion, is shut or open, or closed or open, or cannot conduct ions and can conduct ions. And since there are receptors, the different inputs refer to the binding affinities of the ligand, weak being low affinity and strong being high affinity. So this basic pattern of 2 conformations with different outputs and different inputs can be written as a state model in this box, where by convention C refers to the weak binding and non-conducting state, and O refers to the strong binding and conducting state. And these 2 global conformations can interconvert. The circles show a schematic of a cross-section through the extracellular domain at the level of the neurotransmitter binding sites. You can see the 5 subunits are labeled around the central blue pore. The binding sites are shown in red. They are either at alpha delta. They are both at alpha delta and either alpha epsilon or alpha gamma subunit. During the course of development, the embryonic gamma subunit is replaced by adult type epsilon subunit. And we're going to be talking mostly about the alpha delta and alpha epsilon subunit binding sites. And these binding sites can switch from being weak binding in oval form, and the rectangular form indicates high binding, strong binding. Down below show some MC simulations to the courtesy of Tapan Nayak at IIT Delhi, which shows the gate region in the center of the pore, the equator of the pore, being either dry on the left so that the pore cannot conduct ions, or wet. And the structure on the left was courtesy of Ryan Gibbs' lab. It's the PDB file 5KXI. We are interested in nicotinic receptors mainly because they are an excellent model system for allosteric proteins in general. So this list of shown to the right here are all things that we take for granted with every receptor, every ligand-gated ion channel that we study when we see a cell response, and we just assume that they all apply. But these were all hard-won concepts that came from the study of nicotinic acetylcholine receptors. So the concept of a receptor, that agonists have an affinity and an efficacy, all came from nicotinic, were first identified and studied in nicotinic acetylcholine receptors, as were these other concepts that are generally applicable, that receptors desensitize, that they're allosteric proteins, that the response characteristics arise from the details, the kinetics of the state transitions, that the single channels, the unitary single channel response is a rectangular pulse, that was not known at one point, and that the receptors can have intrinsic activity. And to this list, I am going to add efficiency. And I think that applies because it derives some affinity and efficacy, it's going to be generally applicable to all receptors. These other ideas that I will talk about efficiency classes and modes and cascades and a zipper mechanism, that there's less data available on other receptors, but I'm going to speculate that these are going to be generally applicable too.